professor at ETH Zurich and also CMU. Onur's interest is in all aspects of computer architecture, with a special attention on memory and storage systems and bioinformatics. Now, Onur is ranked #1 on ISCA Hall of Fame, #2 on HPCA Hall of Fame, and I think #3 in Micro Hall of Fame.
That's not in my bio.
Okay, but this says something, right? And given how our community is so small, Onur has about 22,000 citations, which is a great deal in computer architecture, I think, and of course a large number of awards and best paper awards. So welcome, Onur.
Thank you, Len. Thank you for inviting me again to this nice workshop that's close to my heart, as many of you here know. Okay, I'm going to talk about, it's a personal journey. I'm going to talk about accelerating genome analysis and the work we've been doing in this area. I think previous talks gave a nice background, especially built Keynote early on. So let's start. Okay, so this is an overview. I think system design for bioinformatics is a critical problem today. It's going to be important, and hopefully at some point companies will jump on it also. We'll see. Because it has large scientific, medical, societal, and personal implications, and some things maybe we don't know of yet. This talk will be about accelerating a key step in bioinformatics, genome sequence analysis. This is a key step, but it consists of many sub-steps also, and in particular I'm going to focus on read mapping, and I'll discuss why that's an important problem in a little bit. There are many bottlenecks that exist in accessing and manipulating large-scale genomic data, large amounts of genomic data. I think that's true for read mapping, but that's true for other problems as well. So we will cover various recent ideas to accelerate read mapping. It's going to be more personal. It's going to be my personal journey in this field since September 2006. Actually, when we started this, I started this while I was hiking with a collaborator at Mount Rainier while I was at Microsoft Research. And we were thinking about doing things in genome analysis. He's in bioinformatics. I'm in computer architecture. I had no knowledge of bioinformatics at the time. I still don't know enough about bioinformatics. Because when you go to a bioinformatics conference, there's a lot of jargon, and it's very hard to, I think, get to the depth of the insights in bioinformatics. But what we wanted to do, based on what we knew at the time, was to have an embedded device that can perform comprehensive genome analysis within a minute, let's say, real time, quickly. We thought it was important to have this device. I believe it has applications, like you could answer questions very quickly, like which of these DNAs does this DNA segment match with? What is the likely genetic disposition of this patient to this drug? So it could be used in life-critical situations. Perhaps a doctor can take this device and use it so that they can quickly determine what drug to administer during a life-critical situation. I still believe that this is really important. We'll get back to it. And at that time, we didn't really even have the embedded technology to sequence genomes really fast with small devices. Today, I think, you heard the earlier talk from my student Damla talking about nanopore sequencing devices. I think today we're getting close to that technology. So we believe in the technology, and technology is appearing going forward. So with that background, I'm going to jump into the problem we're tackling. That's a DNA read mapping. I'm going to give you some background on that first. And then we're going to talk about the approaches that we've taken so far. Algorithmic acceleration, and then hardware acceleration. And then I'll hopefully talk about future opportunities and new sequencing technologies. So I'll go through some of these background really fast. Hopefully everybody knows what a genome is. It consists of a bunch of base pairs, ACTGs, and there's a lot of literature on this clearly. And it all starts with the DNA, basically. DNA gets transcribed into RNA, and RNA gets interpreted into proteins through translation. Essentially, these DNA makes up what we are and what we are predisposed to. Of course, it's not the only thing. Clearly, there's a huge nature versus nurture debate in many fields. And this is the nature part. It affects your predisposition to many things. Right? Okay, and this is fun. These things are real. This is actually a really interesting thing because this Henrietta Lacks genome was taken from her without her consent. It actually enabled a lot of progress in the field. So today, actually a big deal in genomic analysis, privacy, but we came to where we are because we actually ignored that privacy issues. It's interesting to think about those things. Okay, so let's go into the meat of the problem. So we want to sequence this DNA that exists, And our goal is to find the complete sequence of ACTGs, basically base pairs in DNAs. The challenge is, unfortunately, there is no machine that takes a long DNA as input and gives the complete sequence as output. All machines are essentially choppers. They chop DNA into small pieces with varying sizes depending on the chopper, but they do not tell you how they fit together. Some technologies give you limited information as to how they're linked, but it's not a complete picture of how they fit together. As a result, you have a computational puzzle. You need to figure out how these small pieces fit together so that you can complete the constructed DNA. I liken this to untangling yarn balls. If you have a cat playing with these yarn balls, ideally you would like to take the entire string of a yarn ball, right, and look at it. But unfortunately, imagine that you don't have the technology to do that. You go through it with scissors and you cut it into pieces, and then you need to construct what this red yarn ball looks like. Actually, the problem is very similar. If you actually take a sample from a colon, for example, you'll get all of these yarn balls from different DNA, different species like bacteria, and you'll need to disentangle what those different fragments are. Bill talked about that in his keynote a little bit. So what I'm going to talk about is not even that sophisticated. I'm going to talk about a single yarn ball and constructing it from the pieces that we've chopped it into. Okay, so then these are the choppers basically. These are not a comprehensive set. We have these choppers today. Some of them are state-of-the-art, very, very expensive, like $3 million. They're very accurate. They produce short reads. Some of them are embedded. This didn't exist when we had our dream in 2007. This is an embedded device. It produces longer reads, but it has a lot of errors in the reads. So you have a trade-off essentially. To overcome those errors, you need to chop many times a single DNA. You need to basically get many samples to be able to build confidence as to what you're looking into. Okay, I'm going to go through this quickly. I think some of these slides have been presented. Basically, this became popular, especially with the Human Genome Project. It's true that there are many, many other genomes, but this is an effort that attracted a lot of attention. It cost a lot of money, as you can see. But now we have reference human genomes, and this is a picture that is a must in all of these presentations. Basically, the cost of DNA sequencing has reduced significantly, especially with the introduction of these high throughput sequencing technologies that I'm going to talk about. That's the big machine that Illumina had that I showed you earlier. Essentially, the cost is reducing much faster than Moore's law is providing benefits to us in terms of the cost reduction of devices. And we're actually, as a result, we're able to sequence many, many genomes in the world. And this is going to increase. Basically, we'll have a huge problem processing these genomes. I believe, basically, if you think about these two curves, we'll probably be able to sequence more genomes than we can process with the technologies. So what is high throughput sequencing? I'm going to go through this relatively quickly again. Basically, people have figured out how to parallelize the sequencing very quickly. This is also called many names, unfortunately, in the bioinformatics area, second generation, next generation, all of these sequencing names. But basically, what we can do is, with very high throughput, we can chop the DNA into small pieces, and we can get the sequences very quickly. So this is an example over here. The sequencer, for example, adds a molecule T to all bases that are near a flow cell surface. You have a glass flow cell surface over here where you send the amplified subsequences, and it observes a chemical reaction. We have a CMOS sensor over here, and if a reaction happens, then it declares the base is A. Of course, there is some error in the process, depending on the technology. And then it does it for A, it does it for C, it does it for G, and then it constructs essentially what it has observed. And then it tells you, okay, this sequence of length 100 consists of whatever it has observed, right? Okay, I'm not going to go into the technology. The technology is also fascinating, but we don't have time to go over that. And there are many other technologies. So these are some of the high throughput parallel sequencing technologies. They basically are what they have in common is that they have small DNA fragments. They first amplify them and they sequence them in parallel. As a result, they do it at very high throughput, very high speed, and relatively low cost. Unfortunately, to be able to work, they have to chop the DNA into small pieces. Otherwise, they get too many errors. So basically, they have low error rates, relatively. It's about 1% or less than 1%. Unfortunately, because they chop the DNA into pieces independently, these reads that you get after sequencing lack information about the order, how they fit together. They're completely independent. So you don't know which part of the genome they're originated from. Now there are some emerging technologies like 10x genomics. They give you partial information about how they're linked together. They're called linked reads, but they don't give you complete information. Okay, so basically we know how to design these machines really well today. They have trade-offs, but we're able to do this really fast. But the next step in genome analysis is to understand what we've got from these devices. So we have all these small fragments. How do they fit together? How do we construct the entire genome out of that? That's going to be what I'm going to be focusing on, read mapping. Why is this important? Because this enables everything else that comes after. So in order to understand what you're dealing with, you need to have this complete sequence. At least you need to have a large enough sequence and know where you're focusing on, so that you can, for example, look at the differences of this genome from a reference genome. You can look at different parts of the genome and say, oh, this gene, you're predisposed to some disease because you have this gene. And then you can do either medical treatment or scientific discovery. So basically, whatever can be enabled over here is limitless, I think. But it all is bottlenecked over here. Okay, so let me give you an example, some example motivating questions. I think Bill talked about many of these motivating questions, but I'll give you a couple more. Some of them are similar. For example, if I give you a bunch of sequences, tell me where they are the same and where they're different. This is a multiple sequence alignment, and then you can compare different species, for example, this way. And you can figure out that humans are eerily similar, whereas humans are different from bananas, for example. I think it's good to put things into context, very amazingly similar to each other, even though we may seem to have huge differences. Okay, another question, given a bunch of short sequences that you have, can you identify the approximate species cluster for genetically unknown organisms? So this is an example of scientific discovery, for example. You've sequenced something out there and you want to know what it's close to. Okay, so the problem is, if you want to answer these questions, you need to construct the entire genome from many reads. And that's the read mapping problem. And unfortunately, today we're bottlenecked when we're doing this read mapping. These are some example numbers. They assume some technologies. They assume, of course, some systems. So we can do the sequencing really fast with these high throughput sequencing machines, but we cannot do the mapping really fast. This is a state-of-the-art mapper, BWA-MAM, executed on a single core. You can, of course, parallelize it, but it takes about 12.5 days. Of course, it depends on what kind of analysis you want to do later and what kind of error tolerance you want to have, which we will also discuss. If you want to, for example, find not exact matches, but matches that are significantly different from a reference genome, then the complexity actually increases. This reduces significantly. We'll talk about that. So before I go into a lot of that detail, let me give you the problem. So basically, if you look at the execution time breakdown of that read mapping, most of it is spent on read verification, which is edit distance computation, read alignment. I'm going to go over that again, but other people went over that. So the problem at hand is we have many short DNA fragments, I'll call them reads. We want to map them to a known reference genome with some differences allowed so that we can construct what we have at hand. Pictorially, we have this physical DNA. We chop it into reads, and then we want to figure out where those reads map to in the reference genome. It's not this nice because the small piece actually maps to many, many locations because it's small, 300 base pair long, and a human DNA is about 3.2 billion base pair long. So you want to actually find out all of the possible locations and then prune them somehow. So as a result, you don't do it once, you do it many times. You actually do amplification. Especially with short reads, you may actually need to do amplification a lot. But this is a trade-off I'm not going to go into. Okay, so basically mapping short reads to a reference genome is challenging because you have millions or billions if you do amplification of 50 to 300 base pair reads. So what are the challenges? Basically, if you want to be comprehensive, we don't want to lose information. Because if you lose information, you don't know what you're losing in scientific discovery. This is going to be the basis of the mapper that we're going to develop in a little bit. We need to find the many mappings of each read. A short read, as I said, may map to many locations, especially with high throughput DNA sequencing technologies. The first key question is, how can you find all mappings efficiently? The second question is, you don't want to find the exact matches of a read. You need to tolerate small variances and errors in each read. Basically, each individual is different. Subject's DNA may slightly differ from the reference because you may have mismatches, insertions, and deletions. how can we efficiently map each read with up to E errors present? And if you really want to be comprehensive, you want to increase that E. And we want to do this very fast, of course. Performance is important. As I said, human DNA is about 3.2 billion base pairs long. And if you do the amplification, you get billions of reads. And state-of-the-art mappers actually take weeks to map a human's DNA, especially if E is high. So how can we design a much higher performance read mapper? So we're going to tackle some of these questions. But it all boils down to this read alignment that was discussed earlier. So the existing mappers spend most of their time in edit distance computation. So what is edit distance? Very quickly, it's defined as the minimum number of edits, insertions, deletions, or substitutions that are needed to make the read exactly match the reference technique. So let's take an example, Netherlands and Switzerland. If you actually want to figure out the edit distance of these, you see that there is one deletion over here, there's one mismatch over here, and there's one insertion over here. If Switzerland is referenced, Netherlands has an insertion over there, right? Okay. So why is it slow? As other talks also discussed, this is done using dynamic programming algorithms. I'm not going to go into this in detail. There are multiple different versions of it. And there's a lot of data dependency when you actually do the dynamic programming in this matrix. Basically, you have to traverse the matrix that way. An entire matrix has to be computed, even though strings may be quite dissimilar. So there's actually potential to improve the dynamic programming algorithms. There's some work in that area, which is interesting, that I'm not going to go into. So if you actually go back to our running example, Netherlands versus Switzerland, you essentially need to, whenever you go through the matrix, you look at 3 of your neighbors, and then this is the dynamic programming table you have. You need to fill the matrix, and then you need to backtrace to figure out the minimum score over here, minimum distance. Okay, I'm going to skip this. This is just an example that shows why is it slow, again, with the running example. Okay, so let me talk about the state-of-the-art read mapper design. There are 2 forms of read mappers. One class is based on hash tables. They're called hash base, seed, and extend. They're also, they use hash tables, suffix arrays, or suffix trees. And the others are based on BWT or FM index-based aligners. So the fundamental difference is if you have a hash table, you index the k-mers in a genome. K-mers are strings that are of length k into a hash table. So you pre-process the genome that you're, the reference that you're going to compare to into a hash table. You can think of it as the index of the genome. And then when you get a read, you search for it and you find the location of a k-mer in the read and extend through alignment. Basically, you do the comparison. I'm going to give you an example of this. So if you actually do this indexing of the genome, it can be more sensitive. You can find all the mapping locations, but it's actually slow. And it requires large memory, but this can be reduced with cost to runtime. The other type of mappers that I'm not going to focus into, but these are actually relatively popular today, use compression methods to compress the genome index, and they're actually good at finding perfect matches. They're very quick, but memory lookup costs actually increase for imperfect matches. I'd be happy to talk about that. As a result, they have reduced sensitivity. They cannot find cases with a lot of high errors, basically. And I think this is good, but we don't know what we're missing in these existing mappers. So that's why we actually focused on developing mappers that are really sensitive. I think this is going to be more important going into the future. But of course, this is a purist distinction. I think you can actually find hybrids of these methods also. Okay, so let's look into these hash table-based mappers that we're going to focus on. So the key idea is twofold. You pre-process the reference into a hash table, and you use the hash table to map the reads. So let's look at the pre-processing. This is based on a mapper that we designed a long time ago. This is the hash table. Essentially, you have these 12 mers in this case. You have strings of length k. And you want to pre-process the reference genome and record where these strings appear, at what locations they appear in the reference genome. So for example, you go through the entire reference genome and figure out all A's appear in these 4 locations. That's part of it. And then you put it into the hash table. And you do it for every k-mer that could potentially be in the hash table. So it's a large hash table. You can calculate the size of it. So you don't want to make it much, this k too large because it occupies a lot of space clearly. Okay, now you can use this index or hash table to map the reads that you get. So you do this pre-processing once for a reference clearly. Now let's see how we use the hash table to map the reads. So let's say we have a 36 base pair read, A's, C's, T's, 12 base pairs each. we chop it into these k-mers, and we index the hash table with each of these k-mers that belong to the read. So let's take a look at the A. So we index the hash table with A's. The hash table says A appears in these 4 locations. And what the mapper does is essentially goes to the first location, location 12, reads a 36 base pair length string from the reference genome, and compares it to the entire read over here. This is called verification. Local alignment is a distance computation essentially. And in this case, they match. Okay, I'll go through this quickly. And the mapper declares that, okay, this is a valid mapping, and it reports it. Okay, let's take a look at 324. In this case, the mapper goes through, accesses the reference genome at location 324. Okay, it gets a string over here. It compares it to the read over here. And in this case, it's a mismatch. Why is the mismatch? Because the first 12 base pairs matched, but the remaining 24, or some number in the remaining 24, didn't match. So we basically did the edit distance computation unnecessarily over here. Right? We didn't really need to do it because it's going to match. So we're going to try to eliminate these invalid mappings. Okay, so basically the mapper goes through all of these locations and then tries to does the edit distance computation in all of those locations, which is expensive. And that was the proposal. So this is actually a good mapper in the sense that it's guaranteed to find all possible mappings that you get of a read in a reference genome. So it's very sensitive, and it can actually tolerate up to E errors. I didn't discuss that, but you can read the paper for that. Unfortunately, it's very slow. And hash table mappers are in general slow, especially if you want to tolerate the errors. The verification and alignment takes too long to execute, and as a result you get the 90%, more than 90% execution time dedicated to edit distance computation. And this is another example of this. Okay, I think I'm going to skip this, but you can imagine what's involved in edit distance computation, right? You need to access the reference genome and you do many base pairwise comparisons. So our goal is to speed up the mapper by reducing the cost of verification. So the overarching key idea that I'm going to develop and give you examples of is to avoid the edit distance computation as much as possible. We would like to filter out very quickly with some information, some meta information, whether a k-mer is going to match a particular location. So we want to filter fast before we align such that we can minimize edit distance computations. And we're going to look at algorithmic approaches first, because when we first started, the mapper is too slow. How do we get rid of these edit distance computations as much as possible? And algorithmically, we have 2 approaches. One is exploiting the structure of the genome. If you actually exploit the structure of the genome, you don't need to do all of these comparisons, as we will see, and then we're going to exploit the SIMD instructions. So basically, the key observation is that most of these verification or edit distance computation calculations are actually unnecessary, because you think that you have a match in that part of the reference genome, but most of them, you do the edit distance computation and you're not going to match, given the number of E errors. Basically, you're going to exceed E errors when you actually do that comparison. It turns out 1 out of 1,000 potential locations actually passes the verification process. So we're doing 999 out of 1,000 uses comparisons, edit distance computations. So you can get rid of these unnecessary verification calculations by doing filtering, which is detecting and rejecting early the invalid mappings. I'm going to give you an example of this. And also reducing the number of potential mappings that you're examining. And we're going to exploit the structure of the hash table as well as the reference genome. So the first observation is that if you actually, when you're actually chopping the read into smaller k-mers, the k-mers that you're indexing the hash table with are actually adjacent in the read. They should also be adjacent to the reference genome. If this invariant doesn't hold, you can quickly say, okay, I'm not going to compare, do the edit distance computation. I'm going to give you an example of this. And read mapper, as a result, can quickly reject mappings that do not satisfy this property. Of course, there's complication. Whenever you have errors, it's not a perfect match. You need to actually be careful as to how you do this. The second observation is that whenever you get a read, how do you chop it into k-mers, and which k-mers do you use to index the hash table with? This matters because different k-mers, some k-mers are cheaper, some k-mers are more expensive. This means that not all k-length strings appear at the same frequency across the reference genome. So we want to exploit that. We want to only choose those k-mers that are cheap. Okay, so I'm going to get to this in a little bit. Basically, read mapper can choose the cheapest k-mers to verify their locations. So there are 2 ideas. One is adjacency filtering. We want to reject obviously invalid mapping locations at early stage to avoid unnecessary verification. So let's take a look at this. So how do we do that? The key insight is that for a valid mapping, adjacent k-mers, strings of length k in the read, are also adjacent in the reference genome. If this doesn't hold, you don't need to do the edit distance computation. So let's take a look at this example. We have this read that I showed you earlier. A is mapped to these locations that are indicated by the red arrows. C is mapped to locations that are indicated by the yellow arrows. And T is mapped to locations that are indicated by the green arrows. Read is like this. You would expect the three k-mers to be adjacent in the reference genome also. So if you look over here, you can tell that this is potentially a valid mapping. But if the k-mers are too far away from each other, like this, you can probably guess that they're invalid mappings. So you don't need to do the distance computations over there, because the distance between the k-mers are greater than the error tolerance that you're looking for, E. So this way you can eliminate a lot of invalid mappings. So basically you want to search for adjacent locations in the k-mers location lists. If more than E k-mers fail, there must be more than E errors. As a result, you get an invalid mapping. I'll give you an example of this very quickly without the errors. Errors complicate the process, you need to handle that. Okay, so let's take a look at this k-mer all A's. We chop the read into three k-mers, we get all A's. We first look at the location lists of A, we see 12. So we would expect the next k-mer, all C's, in the hash table to have a location list 24 if the entire read is actually present in the reference genome at that location. So next we go through, we check that expectation basically. We check the location list of all C's and ask the question, is 24 there? If it's there, that's good. Now we go and check the next k-mer. So that should be at 36 because this is 12 base pairs. Is that true? That's true. Then we have some confidence that the three k-mers that are adjacent in the read are also adjacent in the reference genome because the hash table tells us that they're at locations 12, 24, and 36. Of course, with errors, you cannot be certain, so you have to go and access the reference genome, get the 36 base pair, and do the edit distance computation to be certain of this. So this is a mapping that's thought to be valid by the filtering algorithm. So okay, let's take a look at the common case. The common case is that the A's are present in location 324. The next question we ask is it present in 336? Is the next k-mer, all C's, present in 336? So you go to the location list, you don't find 336 or anything close to it in the location list. So you can quickly eliminate this location and you don't need to do that distance computation. And most of the cases are like this actually. So basically the mapper operates by going through the location list and trying to match the locations of adjacent k-mers in the reference genome. Okay, you keep doing that. I'm not going to go into the detail. But is there, most of the, essentially most of the locations are not examined because of this filtering mechanism. Okay. Okay, let me give you the other idea, which is orthogonal to this, which is cheap k-mer selection. Basically, we want to reduce the absolute number of potential mapping locations to actually verify. So what's the idea? So the key insight is that k-mers have different cost to examine. Cost meaning that they map to different number of locations. Some k-mers are cheaper because they have fewer locations in their location list than others. They occur less frequently in the reference genome. So we want to sort the k-mers based on their number of locations and select the k-mers with the fewest number of locations to verify. And if you are confused about k-mers, think of them as k-length strings or substrings. Okay, so this is our read. In this case, I extended it a little bit. Our error tolerance is 2. We want to examine 3 k-mers based on the pigeonhole principle. Basically, you want to select 3 k-mers that are the cheapest. So let's take a look at, if you select this k-mer that looks like this, it occurs in 4 locations based on the hash table that we built in a pre-processed way. And you know how many locations each k-mer occurs at in the reference genome. So if you look at these red ones, they occur at 1K, 2K, 1K locations. So those are expensive k-mers. You don't want to go through all of these reference lists. If you actually choose these 3 k-mers, you need to examine, let's assume the baseline. Okay, baseline previous work actually doesn't care about this, so it could potentially choose 3004 locations. Basically, these two expensive k-mers and this one. So if you're choosing every other k-mer, you would basically need to examine 3,004 locations. But if you actually choose the cheapest k-mers, you just need to examine 8 locations. This is okay, of course, without using the adjacency filtering. Adjacency filtering and cheap k-mer selection have some interaction, of course, right? So the key idea is to choose these k-mers and access the hash table only with those k-mers to see if the read actually are in the locations that are in those location lists. Okay, so we implement this in software based on in our mapper and create a new version, and we test it with real read sets that are public. You can actually replicate these experiments if you're interested. And I'll give you the results very quickly. Essentially, this way we can speed up the mapper up to 19x. Of course, it depends on the input set, different genomes. And the reason we can speed up the mapper is because we actually eliminate about 99% of these edit distance computations by taking advantage of the structure of the reference genome. Okay. Okay, so the conclusion is we have read mappers that perform poorly in mapping millions of short reads to the reference genome in the presence of errors especially. The observation is that most of the verification calculations are unnecessary, so filter them out. We exploit the structure of the human genome to reject invalid mappings early, that's adjacency filtering, and we reduce the number of possible read mappings to examine using this cheap k-mer selection, selecting the cheap k-mers only. And the key takeaway over here is to exploit the structure of the human genome, and that's enabled by indexing the human genome with pre-processing. I say human genome, but it's really any genome. So as a result, you can obtain about 20x, 19x speed up over the state-of-the-art mapper without losing valid mappings. So if you're interested, you can actually download this code. This is not the state-of-the-art at the moment. I'm going to get to the state-of-the-art in a little bit. So after this, we wanted to actually look at optimizing the algorithms using the SIMD hardware. And I'm going to give you an idea related to that actually lead to hardware acceleration mechanisms afterwards as well. And that's the idea of shifted Hamming distance. Let me give you the key observation and the results very quickly, and then we'll go into the key idea. So if two strings actually differ by some number of edits, let's call them E, then every base pair match can be aligned in at most 2 E shifts. I'm going to give you what this means in a little bit, by shifting one of the strings. Why? Because if you actually shift the string by 1, you're really correcting for one error, either an insertion or a deletion. So basically based on this, we developed the metric of shifted Hamming distance. It's essentially an AND of 2E Hamming distances of 2 strings. And if you actually compute this metric, you can quickly filter out invalid mappings. You cannot be sure that the mapping strings actually match each other, unless you get a perfect match, of course. But you can use this to filter out the mappings. And you can actually use the bit parallel operations that might nicely map to SIMD instructions. So the key result that we have over here is you can get 3x speed up compared to the best implementation that was from Gene Myers. He had a bit vector algorithm with a very small false positive rate. So this is a filter again. You're not doing alignment completely. We're filtering invalid mappings. And this is the fastest CPU-based pre-alignment mechanism that we know of still. So okay, let's take a look at an example. I picked one of my favorite cities over here. And what is Hamming distance? Basically, Hamming distance compares this string to this other string. Let's assume that this is the reference and this is the query that you're comparing to. So Hamming distance clearly, in this case, gives you 8 matches and 0 mismatches. But let's say our query was a little bit different. It has a deletion. For whatever reason, somebody misspelled it, right? So you have this deletion. If you actually do the Hamming distance over here, you'll get 3 matches and 5 mismatches. Not good, because these strings are actually very similar to each other, right? But we want to get the benefit of Hamming distance, which is really simple computation, XORs, while also figuring out that these strings are actually similar to each other. So how do we do that? And the key insight is that there is a deletion over here. And you can discover that there is potentially a deletion by shifting the string to the right by 1 and doing the comparison again. So let's take a look at what we do. Basically, shifting a string helps similarity search. This is the reference. This is the query string. We do the Hamming distance compare with here. Of course, you need to do the padding. So we get 3 matches and 5 mismatches over here. And then we shift the string to the right by 1. What do we get over here? In this case, we get 4 matches over here and 3 mismatches over here. But now what we can do is, okay, I'm going to do this again over here. Now you have 3 matches in this non-shifted version and 4 matches in the shifted version, you get to this by XORing. So if you actually XOR this one with the non-shifted version, you get 0s over here and 1s over here. You shift the query by 1 to the right, you get this, and you XOR it with the reference string. You get 4 zeros over here and 3 1s over here. And then now if you add these two masks, XOR masks, what you get is actually only this location is different, and you actually have 7 matches over here. And that's the idea. So let me, basically what we've done is we actually parallelized the comparisons that you have. We create, if you go back to the dynamic programming matrix, we have these 2 deletion Hamming masks over here, and then these 2 insertion Hamming masks over here, and then you can compute them completely in parallel without any data dependencies. So let's take a look at how this works. So I'm going to, let's assume that this is our query and this is our reference. This is the Hamming mask of them without any shifting. And we shift it to the right, we shift the query to the right by 1 and do the XOR. That gives us a 1 deletion mask. We shift the query to the right by 2, do the XOR, that gives us a 2 deletion mask. And then by 3, do the XOR, that gives us a 3 deletion mask. This way you can tolerate 3 errors, right? And then we shift it to the left also to find out the insertions. So you do 3 insertion masks also with 3 XORs and 3 shifts. And then basically these are the zeros, which means that these are the parts that actually match. And you want to find the longest strings of zeros in this case. So if you actually AND these, what you will get is something like this. But we have another process over here to eliminate some spurious zeros, which I'd be happy to talk about. Sometimes you get spurious zeros because of the ANDing operation over here. Everything becomes 0, right? But you may not actually have a 0 over there. Anyway, so basically by XORing the query and the reference, shifted versions of the query and the reference, and then ANDing the results, you get this AND mask at the end. And what this AND mask tells you is, if you have 1s over here, those are the parts that are potentially different. Now if you get too many 1s, then it means that the strings are very different from each other. So you compare the number of 1s that you have in this AND mask to a threshold. If the number of 1s is greater than the threshold, you reject the mapping, saying that these are too different from each other. If the number of 1s is less than some threshold, in this case it's 3, the strings look very similar to each other, so you'd better do the edit distance computation. That's the idea. And you need to pick the threshold such that you don't filter out correct mappings later. And that's the idea. Okay, so if you actually think about it from the dynamic programming matrix terms, as opposed to computing these scores in the dynamic programming with a lot of data dependencies, what we've done is we actually compare the shifted versions of the strings, and we don't have any data dependencies. We're basically looking at comparisons across these diagonals, and these diagonals can be computed independently in this case, and then you need to do the ANDs afterwards. So this is very suitable for acceleration with anything that can do fast bitwise operations clearly. So we're going to look at SIMD operations, and we did with the SIMD operation, and we got the result that this is 3x faster than the fastest previous filter, which is Gene Meyer's filter, which is actually a beautiful paper that was written, I think, in 1990s. Okay, so this becomes a new bottleneck right now. So we have, if you take a step back, sequencing generates many reads, each of which potentially map to many locations. Now we do the filtering, we also call pre-alignment. It eliminates the need to verify or align the read to invalid mapping locations. And if you do it well, you get rid of a lot of potential alignments. And you do the alignment only on those things, on those reads that pass the filter, because this is costly. And after you do this, the new bottleneck in read mapping becomes a filtering step, pre-alignment step. And that's why we want to accelerate it next. So how do we accelerate it? Clearly there could be algorithmic approaches to accelerate it, which is very interesting, but we're going to look at hardware acceleration at this point. So we're going to look at specialized architectures first, FPGAs, and then processing in memory. Okay, I've already said this, but I would like to point out one thing over here. Modern read mappers reduce the time spent on alignment for increased performance. This can be done in 2 ways. You can optimize the algorithm for alignment, actually. You can optimize the edit distance computation. I briefly told that. There is a lot of work in this area. People actually built a lot of FPGA accelerators to optimize the alignment, which is very interesting, and I think there needs to be more work in this area also. We're taking a different approach. We're not optimizing this. We want to get rid of this as much as possible. So we want to reduce the number of alignments necessary by filtering out mismatches quickly. Both methods are used by mappers today, but filtering has replaced the alignment as the main bottleneck. Now we're going to try to improve the filtering step with hardware acceleration. So how do we actually improve it? So ideal filtering algorithm actually should not filter out any correct mappings, clearly, and should filter out all incorrect mappings. And if you actually do that, you don't have a filtering algorithm anymore, that's an alignment algorithm. So that's great. So usually these are not perfect, but you want to actually get as close to these ideal as possible. Of course, while you're doing that, the filtering algorithm should be much faster than the mapper itself. Because if you actually spend a lot of time on filtering, you may actually slow down your entire mapper, even though your alignment cost may be reduced. And as I said, we've reduced the filtering to these shifted Hamming distance computations that are done essentially in a bit parallel manner. So the next step we took was to actually put that into an FPGA. I'm not going to go into the details of how we designed the hardware, but you can imagine that it's not that hard. This is the algorithm that I showed you. Basically, in the FPGA, we have these processing units called gatekeeper processing units. You have shifted versions of the reads, to the right shifted and left shifted version of the reads. And then these units can do XOR operations, and then they use a lookup table to figure out what is your shifted Hamming distance in the end. And we can do this in parallel. You can get the entire read and do these computations in parallel. And this is what the FPGA in the end looks like. We have these processing cores that are essentially what I showed you over here, and they operate in parallel. And there are no data dependencies here, so this is actually relatively simple. And you can actually utilize most of your FPGA space to do this in parallel. So we get significant data throughput, 13 billion bases per second. And of course, this depends on the errors you have, ease you have, and how many mappings you want, and also the length of the reads that you have. Okay, I'm going to skip this one over here. So what is the benefit that you get with this? You get about 2 orders of magnitude faster filter. That's just a filter. You also get lower false accept rate because we optimize the algorithm a little bit, and you get about an order of magnitude speed up in the entire read map. And this is also available online. You can download it and take a look at it. So basically our conclusion with this work is FPGA-based pre-aligned greatly speeds up read mapping. You get significant speed up over the state-of-the-art mapper, and it can be integrated actually with the sequencer. It can help to hide the complexity and the details of the FPGA if you actually do this in the sequencer itself. And I think this can enable real-time filtering while sequencing. So if you're interested, you can try and look at the source code. I think several people tried it and they've seen that this works. I'm going to skip this. You can actually do algorithmic improvements to make it more accurate. So one of the issues with this sort of filter, shifted Hamming distances, when you're doing the AND, you lose a lot of information, especially if you want to tolerate a lot of errors, and actually gets rid of a lot of information you have. And it's not a nice operation. If you do counting, actually, it would be better, but of course it's more expensive to do the counting. So in this case, we actually develop an algorithm that uses divide and conquer methods to get better accuracy in the filtering. So this is a much more accurate filter, but its performance is slower. So there's a trade-off between how fast your filter should be and how accurate your filter should be. By changing the parameters of your algorithm, you can actually get a better read mapper in the end. And this Accelerator is also suitable for an FPGA. You can read the paper for results. Okay, let me get to the latest part of the talk. So what is the problem? Actually, all of these algorithms are heavily bottlenecked by data movement, especially if you optimize them. So we see this in the gatekeeper. FPGA performance is limited by DRAM bandwidth. That's true for shifted Hamming distance. That's also limited by DRAM. So we've been examining processing in memory solutions to alleviate this bottleneck. However, of course, you need to design mapping and filtering algorithms to fit processing in memory. I'll give you an example of this quickly. Later, Jeremy will give the full talk related to this. So essentially, we're using hash tables. This is a summary of what we're doing. We're trying to accelerate the filtering step. So this gives you an example of the filtering. So filtering gives you a match or a mismatch at the end. I'm going to skip that. So we need to design mapping and filtering algorithms that fit processing in memory. How do we do this? So we're going to have a completely different representation of the genome so that we can do the filtering efficiently in memory. I'm going to introduce some data structures, bins and bit vectors, how do we check a bin, and how to integrate what we call GWIM filter into a mapper. So what are bins? Basically, we partition the genome into large sequences. In this case, they're called bins. So imagine a genome that looks like this. You partition into bins. And you represent each bin with a bit vector that holds the occurrence of all permutations of a small string, let's call it a token, in the bin. So let's take a look at the bit vector. Basically have these 5 Murray tokens, five length tokens. And you basically represent whether these 5 length tokens exist in the bin. So you take this part of the reference genome, you basically go through it and say, oh, all A's exist in bin X. All CCCCT doesn't exist in bin X. So you form these bins by pre-processing the genome. And to account for matches that straddle the bins, you need to employ overlapping bins, because otherwise you will lose information, and information loss is not good. A read now completely falls within a single bin. So basically, by pre-processing the genome, you form these bit vectors. This is an example of this bin over here and what the bit vector looks like over here. Essentially, it looks like this. This may not be a good example over here. So you go through the entire reference genome, you create a single bit vector for each bin over here, and the bins are overlapping. So you have bin 1 over here, bin 2 over here, bin 3 over here, and basically the bit vectors represent whether the 5 length string actually occurs in that part of the reference genome. Now we can actually search this in parallel. And you can do the calculations, and you can size your bins and size these bit vectors such that you get a memory footprint that's reasonable depending on where you're going to execute this in memory. So let's take a look at how do we check a bit. Basically, we want to determine whether we want to discard potential match location in a given bin prior to alignment. So let's assume that this is our read sequence. Now we chop it into tokens of length 5. And these are the tokens that we get. And then we read the bit vector for each bin number. So this is the bin number, bit vector for bin number X over here, and then we check whether the tokens actually exist in that bin. So it's basically accessing that bin and getting results as to how many of these actually exist. So you count the number of ones over here, sum them up, and compare it to a threshold. If enough of these tokens actually exist in that bin, that gives you a reason to go into that bin and do the alignment. And hopefully you've selected your parameters such that you minimize the probability that you're going to get false positives in this process. Okay, so that's the idea. If the threshold says, if the comparison says this is not greater than the threshold, you don't do the edit distance comparison. Otherwise, you do the edit distance comparison. But now we need to integrate this filter into a mapper. Hopefully after this it's relatively easy. Somebody needs to give you the potential locations that you have the seeds. And you get the read sequence, and this grim filter tells you, oh, these are the possible places where this read sequence may actually exist. And then it sends it to the mapper, and the mapper checks only those locations, potential seed locations, that are predicted to match. I went through this really quickly, but this is a relatively easy part. Once you know whether, once you know if the filter tells you that this particular read potentially exists in this bin, then you do the comparison. Okay, this way actually you can eliminate a lot of the edit distance comparisons. But this is a completely different algorithm, clearly, that's designed to have simple operations. To check a given bin, you find the sum of all bits corresponding to each token in the read, and you compare against the threshold to determine whether to align. These are very simple operations. And each bin is operated on independently, and there are many, many bins. And this is also memory bound because you have frequent accesses to the large bit vectors. You can optimize it, of course. And we actually designed this to fit in-memory processing. You could do this in SRAM if you actually fit all of the bins in your SRAMs, but we looked at 3D stacked DRAM over here. So we're going to take advantage of a logic layer in the 3D stacked DRAM to actually do these kind of calculations. And 3D stacked DRAM exists, it's already there, but we want to use it for processing in memory. I'm going to skip this relatively quickly because we don't have a lot of time. But basically we're going to take advantage of the high bandwidth in the logic layer. And to be able to do that, we need to appropriately distribute the bit vectors throughout the memory. So how do we do that is the key over here, actually. Let me go to that. So we want to lay out the bit vectors in a bank such that we can process many bit vectors in parallel. This is how we lay out the bit vector. So this is the bit vector for bin 0 over here, and then bin 1 over here, bin 2 over here. We lay them out in the columns of the bank. And the rows essentially tell you which token that you're searching for. So if you want to search for token all A's, you can do it across many bins that are mapped to that bank. So if you're searching for token all A's, you can actually do it completely in parallel in all of the banks. Actually, if you actually stretch it, you do it in all of the subarrays that you have in the DRAM. And then you get an answer whether A actually, in which bins A exists, all A's exist. And this enables filtering many bins in parallel. And if you look at what kind of logic you need to add underneath, it's very simple. Basically, per bin, this is a per bin logic module. You need to have a comparator and an accumulator that increments how many of these ones that you see. Okay, and then essentially you go through you go through each token that you have in the read, and you accumulate the number, and then you compare the number that you get over here to a threshold. If the threshold passes, then that says that you need to do the edit distance comparison. Otherwise, you don't do the edit distance computation. Okay, so this is low area overhead and simple implementation. Jeremy will talk about this more later on, but it essentially doesn't consume a lot of space. And this was published recently last year. I'd be happy, Jeremy will give that talk in full. So what is the performance benefit that you get? So we test this with real data sets again from the 1000 Genomes project. And we evaluated both performance and false negative rate. This is an algorithmic change that it actually reduces the false negative rate significantly also. And we compare it against the state-of-the-art filter. So the performance results, it's about 2x performance improvement overall. I think there's room for improvement over here. I believe you can get more by optimizing some of the thresholds that we have. But we didn't do that in this case. But it's still substantial. And it's actually a better filter from an algorithmic perspective because by doing this binning, you actually reduce the false negative rate significantly. Okay, if you're interested, that's the paper. I think I'm going to, how much time do we have? Okay. So let me go to the emerging technologies. I was going to talk about graphs, but I think there's been talk about graphs, so I'm going to skip that. I think graphs are actually important, as Bill and other talks mentioned. Things are moving more into graphs in terms of representation of the genomes, and graph crossing is becoming more important in bioinformatics. But instead of talking about graphs, let me talk about future opportunities, new sequencing technologies. Damla talked about this a little bit. I'll give you a similar but slightly different perspective. Essentially, genome sequence analysis has become very interesting because of high throughput sequencing technologies. The downside of high throughput the upside is you get very high throughput, very low cost, but you get these short reads and reads lack information. So it's a computational bottleneck. Nanopore sequencing is an emerging technology. It's actually used right now, and it's produced. It's inexpensive, it has long read lengths, it's portable, it produces data in real time. So it has all the properties that we want when we actually dreamed about having this embedded device. Except it has a problem which we will discuss. Essentially, you can plug it into your computer right now. People are using this in Africa, for example, to sequence different viruses and understanding them. But unfortunately, because they don't have good computation capability to do the sequence analysis, they're sending all the data into some data center in the US or Europe. So they're exercising all the data movement bottlenecks that we have in all systems today. What we want to do is actually do the embedded computation embedded into the device and minimizing the data movement such that you can actually get the results immediately. So Damla gave you an introduction of this. Essentially, nanopore is a nanosequel hole, and you pass ionic current through the nanopores in nanopore sequencers. And when a DNA strand passes through this nanoscale hole, the sequencer measures the change in the current, and different bases lead to different changes in the current because of the different electrochemical structures. As a result, you can detect whether you have an ACTG. Right? So this technology has a lot of advantages. It's very interesting, and people are developing new technologies also. It allows sequencing of very long reads because you can actually pass very long reads through that sequencer. It provides portability, low cost, and high throughput. I already said that. The major drawback in this technology is high error rates. Essentially, we do not know exactly how to reduce the error rate. You get about 10 to 15% in the best case error rate. So you actually need to do the sequencing many, many times. So you still have a computational bottleneck here, even though you have long reads. Sequence analysis tools have a critical role to overcome these high error rates. That's why we did the study that Damla presented, because we want to take better advantage of the technology and understand its bottlenecks. And faster tools are still critically needed in this space, I think, so that we can take advantage of the real-time data production capability. This is actually something that can enable real-time analysis of genomes. As Damla presented, this is actually the nanopore genome assembly pipeline. You basically need to call the bases first, understand whether they're ACTGs, and this turns out to be the bottleneck in the current tools. We'll see if that's solved. But there are bottlenecks all over. I still believe read mapping is a big bottleneck once this is solved. That's why I think read mapping is very fundamental over here. And if you look at the paper gives you a lot of detail on the combinations of tools, because some combinations make sense, some combinations actually don't make sense. And there's not enough understanding in the field about which tools should be matched with which other ones. And this is a moving target right now because people are clearly generating new tools for each step. And we need to somehow understand how we actually form the entire pipeline in a much better manner, as opposed to optimizing maybe some individual tools. Okay, I'm going to skip those. But if you're interested, I'd be happy to talk about the paper in detail, and Damla is also here. But this is one example of sequencing technology that I think can potentially revolutionize how we actually do genome sequence analysis, because it's really embedded. It's very different from the other sequencing technologies that have other downsides, like very short reads. Okay, so that's basically almost the end. I'll conclude with what I said early on. I believe system design for bioinformatics is a critical problem. Of course, this is huge, right? Bioinformatics is huge. We've just focused on genome, but bioinformatics is much greater than genomes. It has large scientific, medical, societal, and personal implications. And I've given you an example of a key step in bioinformatics, genome sequence analysis. And even within that example, I focused on a particular step, read mapping, and how to accelerate it. And we covered various ideas to accelerate read mapping, both algorithmic approaches as well as hardware acceleration approaches. And I think there needs to be more work in both areas to actually get this to the state that we want to. Because I don't think we're there. Recall our dream. Our dream was actually to do this within a minute, for example. Of course, this is a very vague goal in the sense that what kind of analysis you want within a minute, right? Let's assume you want to do the most complicated analysis. Clearly, we're not able to do it within a minute right now because we're bottlenecked by the sequencer, but we're bottlenecked by computation more than the sequencer, actually. So that's why there are many future opportunities exist, especially with these new sequencing technologies, and especially with the applications and use cases. If we can somehow embed the application into this embedded device, we could actually achieve even more. Okay, with that I acknowledge all people who contribute to it and people who educated me in bioinformatics, some of them over here. And all papers that I mentioned, source code, everything is available online. You can download them, test them. If you have questions, you can ask us. We'd be happy to help. Thank you.
We have several minutes for questions, so go ahead.
Yes.
So actually regarding nanopore, I had a question. So when I've spoken to people in the field, they believe that it has a lot of systematic issues, like systematic errors which do not get corrected by consensus. Have you tried to explore that? Like I saw the review paper.
Yeah.
But have you seen how the quality varies with coverage, for example?
Yeah, definitely there are systematic errors, and we haven't really tried to improve the algorithms yet. That's a step that we're looking into right now. I believe those are the directions that should be examined to actually make the technology viable. Get rid of the systematic errors as much as possible. But that's a very good point. Yes?
Maybe it's too much to ask this question. So in the grim filter, you mentioned that in order to search a camera or something, you can activate all the banks in the memory, or you can even go further to activate go subarray level parallelism. Mm-hmm. Does that incur a lot of energy consumption?
Yeah, certainly. I mean, in the best case, you would like to actually do it across all of the subarrays, if possible, right. And you're limited by power. Yeah. Which is related to reliability issues. So you're limited by potential reliability issues and power. It will increase your energy, but processing in memory will come at a cost. So if you have active memory that's doing operations, you cannot keep it. You need to find a way of keeping it cool, basically.
Okay.
That's true for all processing in memory approaches, by the way, not just this particular processing in memory approach.
So how important it is to evaluate the energy cost of processing memory?
I think it's very important, and there definitely needs to be more work done in that area.
Okay.
No question about that. We didn't evaluate in this particular case because we wanted to focus on the potential in terms of performance. And we assumed that you could actually do these because it's very simple logic. You just need to increase your power budget such that you need to cool the device. Yes, please.
So, as Bill actually mentioned, the problem with hash tables is that you can't really parallelize them. So it looks like in green filter, you went the other way.
That's right.
You did parallelize things.
That's right, exactly.
Okay, so that
So
was
grim
one-
filter is essentially a different approach. It gets rid of the hash table, as you've seen. It basically represents things in a different way, such that it's bit vectors.
So it's more of a brute force approach, but it can be easily parallelized.
Exactly, exactly. And it fits a lot of the other approaches. So we haven't looked at grim filter on other devices, but maybe Jeremy can talk about that more.
Right.
I think there is benefit to hash tables because they give you a very high comprehensiveness, right? In terms of if you want to actually tolerate, find all of the mappings.
Unless
There
it's
is
de
benefit.
novo, of course.
Unless it's where?
Unless it's de novo assembly.
Unless it's de novo assembly. That's right, of course, yes. Of course, you cannot pre-process something that doesn't exist, right?
Actually, when we do de novo, we don't call them hash tables, we just call them seed tables, so we don't hash, they're direct lookups.
Okay.
There's no collisions, there's no misses.
Direct lookup.
And we actually, when we do de novo, we take all of the reads and we treat it like a reference genome and we index it and build that table. And then we take each of the reads against that to find where the overlaps are.
So you have to index every read
Yeah.
for that.
We actually concatenate them all together
Right.
and then index that.
So like it's one huge counting
Yeah.
and you just.
Yeah. Yeah, so it's kind of similar.
But this has to be done for in every case, like.
Yeah, but
Not-
it's not
not
the
like
rate
the-
limiting step. Actually, the alignment is still the rate limiting step.
Okay. Right. Okay. Thank you.
Okay, thank you, Onur.